12th Biology, Chapter 5, Molecular Genetics. In this part, we are going to discuss about packaging of DNA helix. Where is DNA located? DNA is a long chain which is located inside the cell. DNA is located inside the nucleus of a cell. The length of the DNA double helix is far greater than the dimension of a typical mammalian nucleus, approximately 10 power minus 6 meter. The size of the dimension of mammalian nucleus is 10 power minus 6 meter, but the length of DNA is greater than the dimension of nucleus. How to calculate the length of DNA? Let us see now. The length of DNA is greater than the dimension of nucleus. Now let us see how to calculate the length of DNA. If we want to calculate the length of DNA, we must know about two things. The first one is the total number of base pairs, that is the total number of adenine, guanine, thymine, cytosine. The second one is we must know the distance between two nucleotides. The distance between two nucleotides, that is internucleotide distance, is 3.4 angstrom, 0.34 nanometer, or 0.34 into 10 power minus 9 meter. So we know the distance between two nucleotides. If we know the total number of nucleotides, we can easily calculate the total length of DNA. The total length of the double helix DNA is equal to total number of base pairs, that is total number of adenine, guanine, thymine, cytosine, into distance between two consecutive base pairs. If we multiply the total number of base pairs with the distance between two consecutive base pairs, we can get the total length of DNA. The total number of base pairs in a DNA is 6.6 into 10 power 9. The distance between two consecutive base pairs is 0.34 into 10 power minus 9 meter per base pair. If we multiply, we will get 2.2 meter. But what is the actual size of actual dimension of nucleus? 10 power minus 6 meter. Length of DNA 2.2 meter. The length of the DNA double helix is far greater than the dimension of a typical mammalian nucleus. Then how to pack or how to keep the long DNA inside a small nucleus? That is what we are going to discuss in this part. How is such a long DNA polymer packed inside the cell? Let us see in step by step. DNA is a long structure made up of many genes. The DNA coils again and again to form chromosomes. Then the chromosomes are kept inside nucleus. Chromosomes are inside nucleus. The chromosomes are formed by the coiling of the DNA. Because of the coiling of DNA, it is possible to keep a long DNA inside a small nucleus. The DNA coils and form chromosomes. The coiling of DNA and package of DNA inside the prokaryotic cell is easier than eukaryotic cell, because prokaryotic DNA is shorter than eukaryotic DNA. Prokaryotes do not have a nuclear membrane, and prokaryotes also do not have chromatin appearance. In eukaryotes, the DNA combines with histone proteins and form chromatin. E. coli like bacteria, they are prokaryotes. Here the DNA does not combine with histone proteins, DNA does not form a chromatin. Then how is the DNA packed in prokaryotes? In prokaryotes, the DNA is negatively charged, it combines with positively charged proteins, and it is packed, it is coiled. This structure is called a genophore. In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA being negatively charged is held with some proteins that have positive charges in a region called nucleoid. This region is called as nucleoid. The DNA combines with positively charged proteins to form a genophore. Here packaging is a simple process. In eukaryotes like plants and animals, the packaging will be a complex process because they have long DNA. Plants and animals have more DNA than bacteria and must fold this DNA to fit into the cell nucleus. In eukaryotes, chromatin is formed by a series of repeating units called nucleosomes. Nucleosomes are absent in prokaryotes. In eukaryotes, the chromatin is formed by a series of repeating units called as nucleosomes. Here each one is a nucleosome. The nucleosomes are arranged. The repeated nucleosomes form a chromatin in eukaryotes. Now let us discuss how a nucleosome is formed. This is one nucleosome. You know what is octamer? Octa 8. Octamer of core histones. Histones are proteins. 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and 2 molecules of H4 form histones octamer. 8 molecules of each 2. They form a central part, and the histone octamer is wrapped or coiled by a DNA. Central histone octamer, 8 histone molecules. The 8 histone molecules are wrapped by 2 coils of DNA, one coil and second coil. This structure is called nucleosome. This is histone 1. Histone 1 is not a part of nucleosome. The role of histone 1 we will discuss later. Kornberg proposed a model for the nucleosome in which 2 molecules of the 4 histone proteins, each one 2 molecules, H2A2, H2B2, H3, H4, are organized to form a unit of 8 molecules called histone octamer. The histone octamers are covered by 2 coils of DNA. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. Histone octamer and 2 coils of DNA form a structure nucleosome. The repeated units of nucleosome form a structure called as chromatin. The chromatin is inside the nucleus. This is one nucleosome. This is another nucleosome. A typical nucleosome contains 200 base pairs of DNA helix. DNA is coiled on the outside of nucleosome. The DNA forms a coiled around the nucleosome. The length of DNA coiling is 200 base pairs. This is one nucleosome, this is another nucleosome. The two nucleosomes are connected by linker DNA. Neighboring nucleosomes are connected by linker DNA. The DNA makes two complete turns around the histone octamers. And the histone octamers are formed of histone octamers are covered by two complete turns of DNA. The two complete turns of DNA are linked by H1, histone 1, histone 1. The H1 is a linker histone. It connects the two coiling of DNA. This is the yellow color one is histone 1. The two strands of DNA are sealed off by an H1 molecule. What is the role of H1 molecule? It is not a part of histone octamer. Its role is sealing off two strands of DNA like a clip. If the H1 is absent, how the nucleosomes will be? If H1 is absent, the nucleosomes will appear like beads on a string. If H1 is absent, the nucleosomes appear like beads on a string. Chromatin lacking H1 has a beads on a string appearance, in which DNA enters and leaves the nucleosomes at random places. The DNA enters and leaves the nucleosomes at random places. If H1 is present, the H1 of one nucleosome is attracted by H1 of another nucleosome, so further coiling is possible. If H1 is absent, further coiling is not possible. If H1 is present, H1 of one nucleosome is attracted by H1 of another and form a coil structure. Further coiling is possible. H1 of one nucleosome can interact with H1 of the neighboring nucleosomes, resulting in the further folding of the fiber. The fiber means chromatin. Because of the attraction between H1 of neighboring nucleosomes, a solenoid structure is formed. A solenoid structure consists of 6 nucleosomes per turn. This is solenoid. The term is known to you. You would have learned in physics. 6 nucleosomes form a solenoid. Again the solenoids, non-histone proteins. The green color globular structures are non-histone proteins. The non-histone proteins help the further folding, high level folding of the chromatin. Additional set of proteins are required for packaging of chromatin at higher level and are referred to as non-histone chromosomal proteins, NHC. You know what are histone proteins, what are non-histone chromosomal proteins, and what is solenoid. A 6 nucleosome form a structure, solenoid. Further coiling is due to the non-histone chromosomal proteins, NHC. Because of coiling again and again, the chromatin is packed in a small area. DNA is a solenoid and packed about 40 folds, 40 times it forms folding. Now let us see the outline of packaging of DNA. DNA is a double helix. It coils around 8 histone octamers, 1, 2, 3, 4, 5, 6, 7, 8, 8 histone octamers and form a nucleosomes. The nucleosomes are attracted with each other by H1 and form a solenoid. By the presence of non-histone proteins, further coiling takes place and form a chromosome. The chromosomes are inside the nucleus. In this way, DNA is packed. Inside the nucleus, the chromatin has 2 regions. They are euchromatin and heterochromatin. In heterochromatin region, the area is thick and highly stained. Euchromatin regions are thin and they are lightly stained areas. Euchromatin region involves in transcription. What is transcription? DNA is copied into mRNA. Euchromatin involves in transcription. The heterochromatin is found in telomeres, that is the end of the chromosomes, and centromere region of the chromosome. In a typical nucleus, some regions of chromatin are loosely packed, lightly stained, and are referred to as euchromatin. The chromatin that is lightly packed, stained darkly, is heterochromatin, tightly packed. Euchromatin is transcriptionally active and heterochromatin is transcriptionally inactive. The euchromatin part is transcribed. Heterochromatin is not transcribed. Which one is tightly coiled? Heterochromatin is tightly coiled. Heterochromatin is found in telomere and centromere of the chromosome. So we will meet again with another topic.